This is question 8 from the 2017 AS Paper 1. And the question asks, give 3 properties of water that are important in biology. So it wants a property. What is a property? A property is something that you can attribute to something. It's a quality or a characteristic of something. And for water, there are lots. Number 1, it's a metabolite. It's something that's produced from metabolism. Number 2, it's a solvent of many polar molecules. Number 3, it has a high heat capacity. That means you need to give it a lot of energy before it actually begins to heat up. Number 4, it has a large latent heat of evaporation. In other words, when it evaporates, it takes a lot of energy with it. It has cohesion between the molecules due to hydrogen bonds. And again, that's useful if you remember back learning about this at the beginning of AS, where we looked at surface tension and cohesion in the xylem. Now, according to the examiner's report, only 32% of students managed to score all 3 marks on this, which is shocking because it's basically a recall question. Where did students go wrong? Well, they missed out key words, like they wrote, has a latent heat of evaporation instead of a large latent heat of evaporation. Or they would just write heat capacity without stating high heat capacity. At the time, students stated random words such as condensation and hydrolysis without further explanation. So my top tip for this strategy would be to practice saying the words out loud. That way you'll remember the sentence as a whole. You won't end up with hybrids of sentences. By saying the words out loud, you're using the motor skills in your mouth, you're using your ear, and you're using sound and your brain. So you lay down different pathways in your brain and you're much more likely to remember something, even if your family and friends do think you're a bit of a weirdo. The next part of the question is quite a lot of reading, and that's where we're going to take a pen and chunk it up as you read through. A student investigated the effect of different concentrations of sucrose solution on chips cut from a potato. Each chip had the same dimensions. The student weighed each chip at the start, placed each chip in a separate test tube containing 10 cubic centimetres of sucrose solution at different concentrations, left the chips in the sucrose for 24 hours, dried the surface of the chips, and then reweighed them again. The crux of this is a very common question, and there's a set of questions that always follow this experiment. Often they'll ask about things like, why did you dry the surface of the chips and weigh them again? And that would be obviously because there may be different amounts of water on each chip, and water has a mass and therefore would affect the result. It is about different amounts of water on the chip and not just the fact that there's water. If it was water, then it would just be a systematic error. But because there's different amounts of water on each chip, then that would affect the result. For part 3, it asks us to calculate the ratio of final mass to initial mass of the potato chips. It's after part 2, but let's just deal with it now because it's on the paper. Now, the examiner's report said that only 22% of students could actually do this. And the first thing about calculating ratios is we have to make sure we get the ratio the correct way round. It tells us that they want the ratio of final mass to initial mass. So the final mass must come first and the initial mass must come after. A ratio is always expressed as something to 1. So to get that 1, what we do is we divide the small number by itself. So if we take the first example, we've got a final mass of 3.82 and an initial mass of 2.79. Let's divide the smallest number by itself, 2.79, and that gives us 1. Then we have to divide the other side by 2.79 because it's a ratio. So that gives us 1.37 to 1. Sometimes this kind of question is expressed as a percentage increase or decrease in mass. Using a ratio is the other way of doing it. I prefer using a ratio because it means when we plot the graph, all our numbers are positive. And clearly, if there was no change in mass, then the ratio would be 1, because divided by itself, it would be 1. And that would tell us the concentration of the water potential of the plant tissue. This links directly to practical 3 of the AQA required practicals. Let's deal with part 2 and then come back to part 3 with the graph. The student produced the sucrose solutions with different concentrations of a concentrated sucrose solution. Name the method she would have used to produce these sucrose solutions. Well, the method is a dilution series. This can also be called a serial dilution. And you'll notice that this is one of the skills from the CPAC, the Common Practical Assessment Criteria. It doesn't matter what exam board you do, they need you to know what a serial dilution is and how to make a dilution series. If you go to my required practicals playlist, you'll see this being done with proper laboratory equipment and not just diagrams like Francesca uses. So the next part says calculate the ratio of final mass to initial mass of potato chips and plot a suitable graph of your processed data. Well, we've processed our data and it just tells us to express the ratios in table 4 as a single number. For example, 5.26 to 1 would be expressed as 5.26. So our independent variable, the one that stays the same, is the concentration of our sucrose solution, and we're going to place that across the bottom of the graph. We need to make sure that we use at least 2/3 of the graph available, and that fits nicely across the bottom. And then up the side, we count the squares and we can see we've got plenty of room. And in fact, if we start our graph at 0.7, because we only need to go up to a maximum of 1.4, then we can space our information out nice and clearly along the axes. Use the column headings from the table to label my axes, making sure to include the units exactly as they appear in the column headings of the table. And this is a top tip to do this, because ratio doesn't have any units. If it did, it would be included in the column heading of the table. So you're safe to assume that whatever appears in the column heading of the table, you can use on your graph to label the axes. So you got a mark for having the axes the correct way around with a linear scale, i.e. not a log, and with evenly paced scale representing the same number of values as you move along the axes. You got a mark for labelling the axes with mole per decimetre cubed and with ratio. And you've got a mark for correctly plotting the values and drawing a suitable curve. Now the curve can be point to point because it's biology, so you can do it with a ruler, or you can do a smooth curve of best fit. Take care not to make your curve go beyond the points you've plotted. There shouldn't be any extrapolation. There's never a definitive line of best fit, but you can see here that this is a curve of best fit because I've left an equal number of points equidistant from the line as my curve has spun through them, as it's tried to hit as many of the points as possible. And just to point out this backslash mark, it's called a solidus, and we use that to separate units when we're labelling graphs or in column headings. That is the appropriate convention, not brackets. And this -3 here, we've got moles to the decimetre to the -3. That just means per. It means per cubic decimetre. It's shorthand for per, and it doesn't mean minus in the mathematical sense. Okay, moving on to the last part of the question, it says, explain the result for the chip in 0.8 moles per decimetre cubed of sucrose solution. So we need to say that the sucrose solution has a more negative water potential than the potato cytoplasm. It wouldn't be correct to say the sucrose solution has a negative water potential because it's a more negative. It's all about relative to where the water is or is moving to. You can also say it has a lower water potential. And then we need to look back to the question, and it asks us to explain. So that's when we use the word so. So we're going to say, so water moves out of the potato into the sucrose solution by osmosis. So the cells decrease in mass. By adding, so the cells decrease in mass, we take the answer full circle because we're explaining the result for 0.8 moles per decimetre cubed, which is where the potato chip has actually decreased. It's gone below 1, a ratio of 1. According to the examiner's report, the most common problem in answering this question was that students didn't talk about the term water potential. Again, remember, you're an A-level biology student now. You've moved on from GCSE. You need to be showing off that knowledge. So anything that's A-level, anything that's uplift from GCSE, you're including that in your answer. Now, just as an aside, just for fun, just as an extra, I have used the graph and I've copied across from the y-axis to the curve and down to the x-axis. And the reason why I've done this is because this represents where the potato didn't gain or didn't lose any mass. In other words, it was at the same concentration sucrose solution as of the cytoplasm of the potato cell. And this is commonly where the question goes. It wants to know why it didn't increase or decrease in mass. It wants you to recognise that a ratio of 1 represents no change in mass. And with the new specification, often they will ask you how you would use the graph to find that out. And of course, you would talk about moving up to the y-axis, moving across from the y-axis to the curve, down straight to the x-axis and taking the reading from there. And indeed, I'm just looking at what I've done there. Can you spot my deliberate mistake? What I've done is I have gone up in twos, 0.2 to 0.4. So where I've written 0.26, it isn't actually 0.26, is it? It's 0.32. I've done a classic student mistake, so it just shows you how easy it is to do that. That just shows exactly how easy it is to make a mistake, but it also pushes the point that when you think you've finished at the end of an exam, it's definitely the kind of thing you can put on your checklist to go back through and check any readings on graphs. I hope you found the video useful. Please don't forget to subscribe. Hopefully more interesting exam questions coming up soon.